Hello, everyone. My name is Jacob Huang. I'm the Executive Director of the IAS City University. This is summer, so we usually do not have workshop or lecture during the summer. So Professor Smell is very, very brief to have the whole extended workshop during the summer. So anyway, today we have a big show. Very important person.
Oh.
Yeah, Professor Smell is a big guy. So you're pretty big and also very important. And from the record, I see Professor Brian ATHEY, or ATHEY, yeah, is a chair professor and the founding chairman of the Department of Computation Medicine and Bioinformatics in the University of Michigan, Ann Arbor, medical school. And he was the director of academics and clinical informatics for the school. And also serves as a founding co-director of the UM Michigan Institute for Data Science. Yeah. So all is very important. And also Professor ATHEY is a PI of one of the most well-established NIH pre-doctoral training program in bioinformatics in the United States. And he has served PI for many different efforts, such as NIH Visible Human Project, DARPA, I don't know what that is. DARPA, okay. Must be very important. Virtual Surgery Project, and NIH National Center for Integrated Biomedical Informatics. And he's the founder of Michigan Institute for Clinic and Health Research, and also as a director for 11 years I'm trying my best because I have a material science background. Yeah, so it's totally different view. And he was the co-founder and later the CSO of the Smart Foundation, special data integration analysis platform. Okay. And since 2010 to 16, he served as a chair for scientific advisor board of ATHEY Health Company in Ohio, and also associated with the Genetics in Salt Lake City, Utah, and he has consulted extensively for Defense Advanced Research Project, again, DAPRA, and NIH Office of the Director, and NIH Chief Information Officer. Yeah. Okay. Since 2017, this ATHEY has served as advisor for Chinese University of Hong Kong in Shenzhen. Okay, and he is an elected fellow of American College of Medical Informatics. So he's really, really important. And today he's going to share a topic on my notes to be arranged. So we will see that later.
Lucky I arranged it.
Oh, you arranged it. Nice. So let's give a big hand to Professor ATHEY.
Thank you. Thank you very much. God, when you listen to something like that, you know you're old. You know, I'm not a mathematical biologist, but I love mathematics, and I think at the end it would be fun to talk about how we could code up some of this for mathematics. You know, I started as a biophysicist, and we use a lot of mathematics in biophysics. And bioinformatics, which is a field that didn't exist when I started, it's one that I had a small part in creating. You know, it needs mathematics, actually. So it'll be fun to kind of, and there's no math in this talk, although I think there's an opportunity for a lot of math, and I try to point at various places where. Yeah, I'm also a professor of psychiatry, which is going to turn out to be very important. And psychiatrists, how the mind sometimes goes awry, and we have mental conditions like depression or bipolar disease or schizophrenia or other diseases of the mind, and they're very hard diseases, and it's very difficult to help those people. And I've been spending years now trying to figure out how to help people with psychiatric conditions, and I've made a little contribution to that, which I want to share with you a little bit today. And I was just, Michael and I were at the data science, School of Data Science at City University, Hong Kong, retreat this morning, and it was a lot of fun. There's a new School of Data Science here, and there's a Data Science Institute at City University, and they've got an initial faculty of about 16 faculty members. They're going to grow it to 30. We've got a bachelor's program and a master's program started in data science. It's a new field that brings together computer and information science and applied math and statistics and various applications, and it's an important new field. It didn't exist when I went to school, and bioinformatics didn't either. We're required to show any kind of potential outside activities. And notice that this company here is in Hong Kong, and I work in Shenzhen, and so I come in and out of this place a lot, and I love it here. So it's fun to be and learn about City University in Hong Kong. Here's my outline. I'm going to tell you about something called pharmacogenomics. I don't know if anybody's heard of that before, but it's a field that where you can make measurements of various biomarkers, and the genomics is, if you can make genomic measurements, something about your genes that say Professor Smale is different than Michael, is different than me, is different than you. Maybe we respond to drugs differently. I think everybody knows when you take a pill that some people do okay with it and some people get very sick. For example, some people take codeine, which is a pain medication. But many Africans, for example, take codeine, nothing happens. They don't feel any relief of pain whatsoever. The question is why? And it turns out that there are different genes involved that code up for different proteins and ultimately pathways that metabolize these drugs. And if we could figure out a way to measure your genes, then we could maybe assign which drug you should take or not take. And I've been studying that for about 10 years now. I've been making some good progress, and it's been helping people. So I'm going to tell you about that. I'm going to tell you about the first generation try and what we did to help depressed people. We've helped about 1 million of them in the United States, which is a lot of people, but it turns out there are 450 million depressed people in the world, so we got a long way to go. And then there is other diseases. And lithium is a drug that we've been giving for people who have mania and bipolar disease. And I'm trying to work on a test to figure out if you could respond to lithium or not, because if you take it sometimes and you don't respond to it, you can gain a lot of weight, like 2 kilograms a week, or your hair falls out, or your skin changes colors. It's really bad stuff, except if it helps you. And then another drug, valproic acid, is a famous old drug that helps people with epilepsy. It helps, and I'll show you what we've been doing to help people who have their brains are injured. This drug really helps a lot with that, and it's very fascinating. And what I'm doing is figuring out how these drugs work and how to measure the genes to figure out whether you respond to them or not. It's very important, actually. And I'll take it all the way down to where we're at with machine learning and the big data aspects of this. Professor Roman was here, and he was a speaker in your series, and he and I are collaborators and friends. And in 2009, we made this chart where we were trying to understand the different levels from the genome to the proteome to the metabolome to the cell, tissue, organ, and then the whole organism, and then the population. And if we can think of it, Professor Smith, I think if we could think about the mathematics of how to bring these levels together, that would be very useful. It's one way to think about it is within a level, you can have networks within a level that have strong interactions, and then between the levels, maybe there's a lot of weak interactions. Like, for example, DNA itself has strong interactions between the bonds along polyphosphate backbone and a bunch of weak interactions and hydrogen bonds that hold the two strands together. You know, it was actually a famous economist and cognitive psychologist, Herbert A. Simon, who way back at Carnegie Mellon in the 60s and 50s started to think about strong interactions and weak interactions. And you know, if we could mathematically encode the strong interactions between the levels and the weak interactions between the levels and the strong interactions in the level, it would probably be quite useful. And it would be helpful for mathematics. Now, over here you have something called the phenome. From the top down, if you collect all these measurements and look from the top down, that's called the phenome. So if you sum up all the molecular measurements from the bottom up, that's called the molecular phenome. And you can measure that. And that's what bioinformatics really does, is measure the collection of different elements that make up these levels that are unique to an individual. And then some parts of the phenome are what you do to yourself with the environment. I smoke cigarettes, I eat the wrong food or something like this. Some parts of the phenome are temporally organized in time. They're physiologically oriented, and so on. And so this is a nice way, and I'll leave these slides here. This is a nice way to think about how to break down a highly complex phenomenon like a biological phenomenon. It's been helpful, and the National Academy of Sciences and Medicine took that slide over and wrote a whole book about it a couple of years later. It was really quite nice to see that. In my field, which I'm going to describe to you, pharmacogenomics, I'm going to explain the first generation of pharmacogenomics and how that worked, and to date the best actual demonstration of that particular field, the first generation, which actually combined different genes together. And I'll explain to you something called pharmacokinetics and pharmacodynamics, and combine these genetic measurements in such a way and relate that to which drug you can take or not take, and do clinical trials from the top down and make assignments, and we did that. In fact, the clinical trial that we just did was just published a few weeks ago. I'll tell you about that. But what I've been doing for years now is inventing the second and third generation, and I wanted to spend most of my time talking about that. And I studied depression. Turns out, you might know this, people who are depressed are often in pain. You know, we don't really understand why that's true, and pain is turning out to be important in its own right because people take things, analgesics like opioids, to avoid pain. And so there's addiction kind of stuff there. Stress-related disorders in general, cardiovascular disorders, and gastrointestinal disorders. I've, the past 5 years, published papers in the field of pharmacogenomics on all those different areas. Now, depression and bipolar disease. I've mentioned 450 million people in the world have it. #1 cause of disability in the United States and around the world. 2/3 of people who commit suicide are depressed. Actually, the most interesting thing is the third who commit suicide who aren't depressed. That's a separate lecture in and of itself. It's fascinating. And not all these people who kill themselves are depressed. There are different segments of the population, males and females in different ages, that have different susceptibility to these diseases. We're starting to actually understand that quite a bit. We have these, at least in our country, in the US, there's the shootings that are related. And despite the fact that this is such an important problem, and I've been studying this in China as well. I visited the third hospital, the mental hospital at Peking Union Health Sciences Center, the psychiatric hospital in Beijing. I've been to the psychiatric hospitals in Shanghai. There's an incredible one in Shanghai, one of the largest in the world, with 6,000 inpatient beds. In Ann Arbor, we have 12 inpatient beds. Okay? It's a problem here too, as it turns out. Challenges. We still haven't figured out how to really diagnose it. It's very actually kind of, we don't know how to classify these different people with diseases. It turns out that people who have anxiety, or anxious people, get nervous, often have depression. Sometimes anxiety disorder can lead to depression, for example. And sometimes, well, many of these things are what we call spectral. Bipolar disease is certainly like that. Some people have it very, acutely, some people not so much. Just like autism, there's a spectrum. Many of the neurocognitive, neuropsychiatric diseases have a spectral characteristic. We're starting to understand that. One of the things that is really been a problem and something that I've been working to try to solve is making some progress actually, is there's no new drugs. Most of the drugs we give people who have these diseases, came around in the 50s. There's so all around the world with all these pharmaceutical companies, hardly none of the companies is starting to pick up again, are trying to develop drugs to help these people. Because it's been so difficult to figure out how to do it. We're finally figuring out why that is. And however, the drugs that we do have are prescribed. 28% of the United States population, 30%, and I think it's probably higher than that, are on antidepressant drugs. Think about that. A third of the population of the US are on these pills. It's something. I don't know the numbers here in China, but I'm sure they're significant. And there are many what we call adverse events to these drugs. And it's in excess of 100,000 visits to the emergency room in the United States where we have something called acute adverse events. You take the drug, it makes you so sick you have to go to the hospital, you feel like you're going to die. So you'd like to avoid that, actually. Now, this fellow here, Dr. Mrazek, who's an old colleague of mine who passed away in 2013 of glioblastoma multiforme, brain cancer, was the person who developed the first concept of pharmacogenomics in the field of psychiatry, and actually the first real concept of pharmacogenomics in all of clinical medicine. He was a good friend and colleague, and his science started the company in the US called Assurex Health, which I ran for several years, that developed the test that was described in his book. And I'm currently now putting together with some colleagues the second edition of this book, which is a more general book now. Once once you start writing books, you know you're in real trouble. It takes a lot of time. Now, we had a test. We had a very important study back in the 2005, 2010 timeframe called STAR-D. And that was a nationwide study to try to figure out something called treatment resistance. And treatment resistance is actually a term that refers to, a patient comes into the clinic and you give them a medication for their depression, and then they don't, it doesn't help them. So they have to take another drug. So they give them another drug. So, you walk into the clinic and say you're depressed, I think you need Zoloft or iloperidine or some kind of drug, Wellbutrin or something like that. How do I know which drug you should take? Usually, I guess based on some other patient that did well with it, I don't have any measurement to figure out which drug you should take or which one that you should avoid. And there's so many of them, it's actually hard. Well, if you fail that line there, that QID 16, that's a test to see whether you're improving or not. It's a questionnaire. And you can tell by the questionnaire whether you're getting better or not. Well, it turns out that, if you take one drug and then you fail, then you take another drug and then you fail, and you take another drug and you fail, you have treatment resistance. We studied that all around the United States and got the numbers to try to understand, in a semi-quantitative fashion, whether you were going to get well on the drug or not. So the question is, how do we improve this situation? Now, here's the slide that just, it's a little hard to see, especially when you have glasses. It's a list of the drugs over here, and this is the hospitalization rate and how many visits into the hospital in the United States and what kind of adverse events they had. This is a big problem. Okay? Now, here's I'm going to give you the basic ideas here for a second. Okay? Now, the way that drug actually is much more complicated than this. I'll try to explain that later. But for now, there's two basic mechanisms, pharmacodynamics and pharmacokinetics. Actually, the ones that we understand the best are pharmacokinetics. So when you take a medication or you take some toxin into your body, the liver and several other organ sites around your body have a set of enzymes called cytochrome P450s. They're in your liver, they're actually in the lining of your stomach, they're up in your brain, they're in certain tissues, okay? And I'll show you in a minute a list of these cytochrome P450s, and it turns out that each cytochrome P450 metabolizes a different substance. It's encoded by a different set of genes, it's on a different set of area of the chromosome, and it encodes a pathway that metabolizes a different set of substances or a different set of drugs. So any substance that you take in your body that is a foreign substance is metabolized by these liver enzymes primarily. It is cleared from the bloodstream, goes out into your bile and out, into, ultimately is processed through your kidneys, through the urine. Now, there is another set of receptors and transporters and things like this that are in your brain and other tissues that also are modulated by the drugs. Like if you take a selective serotonin reuptake inhibitor, an SSRI, it interacts with a certain receptor that changes the modulation of the axon, when the neurons fire and they send off boutons over to the axon, it changes the rate of that. And so drugs can affect receptors and transporters and things like that in various tissues like the brain, in various brain circuits, as the drugs are metabolized in your liver by the cytochrome P450s. And we can actually start looking at this in more detail. So here's a list of the different cytochrome P450s in your liver. They're actually variants of the same kind of enzyme, but they have significant differences, and they actually metabolize different substances in the liver, in the liver microsomes. And so you have cytochrome P3, 3A4 and 5, 2E6, CYP1A2, 2C19, and so on. And so this is the proportion of the medications that are metabolized by the various cytochrome P450 drugs. So there's about 60 enzymes that are responsible for drug metabolism, and there could be a combination of these enzymes, in fact they are, that metabolizes different drugs and substances. And what you'd like to do is you'd like to make genetic measurements, genotype the different enzyme variants and relate that to how the patients respond to drugs by studying how they do so quantitatively in the clinic, and that's what we do. And so here's the different kinds of drugs that we use to treat the patients. Here, this is the enzyme that, the cytochrome P450, the pharmacokinetic enzyme that metabolizes these drugs. And then, we can actually make a classification, and we can come up with a phenotype from the top down. Do you metabolize ultra rapidly the drug or the substance, or are you a slow or poor metabolizer, or are you somewhere in the middle? Okay, for example, and Michael already knows this because we've gone to dinner a couple times, right? I can drink a double espresso at 10 at night and go right to bed, right? So I am an ultra metabolizer of caffeine. Right? There are other people that we know, if they look at a cup of coffee at 6 at night, they're going to stay up all night. They are a poor metabolizer of caffeine. All right? Drugs is the same way. Or you could be an intermediate metabolizer. And if you think about it, you can think about different things that you eat or take into your body that affects you differently than your friend or your spouse or something like that. We can actually measure that. And, we can measure each cytochrome P450 variant and determine the phenotype. And actually, we can do better than that. We can get microsomes from animals, and we can have the different cytochrome P450s in a test situation in the laboratory, and we can make quantitative measurements and start to make models and system models and things like that. We've done all that. So, and we know which drug for which condition, in which cytochrome, or other important molecule, and yet all this has been studied and tabulated. It's amazing. And so if you think about it, from a computational medicine perspective, we can bring all that data together in a computer. We could make we could get an algorithm, and we could classify how patients respond to different medications. And the first attempt at this was made by Dr. Mrazek. And he had 6, the original test, he had 4 cytochrome P450 variants and 2 these are pharmacokinetic enzymes. These are pharmacodynamic molecules. One's a transporter and one's a receptor. Okay, and there's your phenotype. And then you have, you know, a lookup table with the drugs, relating to the drugs and how the enzymes react. And then you can actually, this is where the color is helping me out. So you can use as a directed. That's supposed to be green, yellow, red. Okay? Like green, yellow, red. Don't take it if it said don't take it if you're a poor metabolizer. Something like that. So green, yellow, red. Use as directed, use with caution, don't take it. Avoid it. Don't take it. Okay? Counterindicated. And you know, I worked for years with a company that started off real small and grew and grew and grew, and put this out on the market to help people. You know, I don't have the slides with me, but I have a slide of mine. It turns out, you can imagine this, right? You take my, cheek swab and measure mine. It's now only 15 genes. The next test I'm going to do is going to have 1,500 genes. But anyway, you swab me and you swab my wife, and guess what? I'm completely different. Every drug that I can take, my wife can't take. We're like opposites. Every one of us is really different. And we can now, in a very primitive way, but in a real way, make this assignment. And the company that we created to do that, we sold, is already genotyped and helped 1 million people find the right medication. It's really helped them. It's just the beginning. It's just one indication. Treatment-resistant depression, major depression. Just think what we could do. We could, and then, when many people in a hospital, like 50, 60 years old, they're on 10 medications. It's called polypharmacy. We can use a computer and figure out how many drugs you should be taking, which ones you can't be taking. It can change the whole way that we work with medications, and that's what we're going to do. And we had a randomized control trial. Here's our little announcement here, and there's our, we did okay, but it's just like, look, we did better than treatment as usual, but not much. So the question is, how can we make this better, and why is it so primitive? We are improving things, but by not much. But it's the best test that has ever been developed. And and the thing about, in our country, you can't actually get paid to do a genetic test for drugs or making any other assignment to help people if you can't figure out whether your test actually saves money. One of the things about genetic testing and all this for medicine is that in the United States, it'll be interesting, it's an interesting discussion about China. Right now, people don't get paid. Actually, we were the first group to get paid for a test that was used for medical treatment. The reason is we did a study to show that people who are depressed don't show up to work, their family loses a lot of money, so if you can get them on the right medication, get them back to work, it saves a bunch of money, and you can actually do a medical economic study. This one was with 2,000 patients. And you can prove to the government that, if you pay me a certain amount for this test, we're actually going to make the economy and the government save money. And that's part of how you do it. The first thing is the basic science, then you have to do the clinical science, then you have to do the economic study if you want to translate a discovery out into the market to help people. It's a lot of work, actually. So you might say, gee, you're doing great. Why, what are you complaining about? Well, it turns out that the test is 15 years old, and you know, I think many of you know Indica. Well, I hired the guy, and you know, I learned a lot from him, and I started to realize that if I apply, now you guys have, many of you heard of his talk, what I'm going to do is I'm going to tell you, I learned about the 4D nucleome, and I translated that into pharmacogenomics and invented a far more powerful test based on Indica's work, okay, over several years. I want to tell you about that. Well, part of it was the so-called missing heritability. You know, genes by genotype, by phenotype. Genotype by environment gives you the phenotype. The genotype, even if it was perfect, can only give you 50% of the story. We weren't getting anywhere near that power, so the question is, where's the rest of the genomic power? Big problem. Maybe it's locked into the 4D nucleome and the epigenome. Okay? So the question is, how do you get it out of there? So I've been really looking at that. You know, and then we only had pharmacokinetic genes, and actually our test that we developed and marketed only was really an adverse avoidance test. This really should be more simple. If you're in the red bin, don't take the drug. You have a lot of adverse events. If I can move you to the green bin, you won't have the adverse events, and you'll feel better. That's very primitive. Is there something better we can do? And can we develop tests for other indications other than this one indication, treatment-resistant depression management process? So there's the 4D nucleome over here. Here's Indica. The 4D nucleome refers to the dynamical relationships among the nuclear structures, the transcriptional landscapes, and the cellular phenotype. That is a good definition. So let's see if we can work with that. Let's see where that can take us. So, here's some background. Well, there's one thing. The brain and the central nervous system more generally is a huge machine, a huge metabolic machine where most of the transcription and most of the gene expression and most of what happens in your body happens right in your brain. So we've got to understand that a lot more, okay? And actually from genome-wide association studies, not only in neuropsychiatric, so the old Francis Crick was a hero of mine. You know, you have the central hypothesis, the central dogma. Genes encode RNA, encodes protein. Everybody learned that, credit school, right? It's likely totally wrong. Turns out that that assumes that what happens, that if you play that, Dr. Mrazek certainly did, is, you know, the hypothesis was that all these different genotypes that the patient had were changing something that was coding the protein of the cytochrome P450. That the cytochrome P450 metabolized the drug differently because there was something that was changing in the protein of the enzyme. That's not really what's going on, actually. It turns out that most of the genetic changes that we measure, we studied this by, and I can tell you what, by downloading every GWAS study, genome-wide association study that was ever done for certain indications, we discovered, and others discovered in other fields, that most of those marks actually don't fall in the protein folding region, but fall outside the protein folding region in something called the regulome. It regulates the gene expression. It doesn't change the coding of the protein. It changes how those genes are expressed or not expressed. Completely different thing. Causal alleles are in the chromatin, in the open chromatin. I'll show you some pictures in a minute. Okay, you have the open chromatin and the closed chromatin. Discovery of humans, you can discover pharmacodynamic networks. Question is how? And the regulatory epigenome is now the foundation of rethinking how we do pharmacodynamic pathways. All right, so here's a picture of the chromatin. You know, the first thing that I did in my PhD thesis was solve the structure of that fiber. I worked on it for 8 years. And then people didn't believe it for 30. But it turns out that that's called the double helical cross-linkers You have a nucleosome and it comes across and it goes like this. It's in the textbooks now. It makes a double helix of nucleosomes. It's called the double helical crossing chromatography. That was my PhD thesis in the 1980s. And I can tell you that it took 30 years to figure out how the chromatin, there's really Adika and, you know, these high C maps and all the work that's been done that's really kind of breaking open how this all works. And one of the important concepts is this topologically associated domain. That in a chromosome, you have chromosome territories in the interphase, and then you have a bunch of topological domains, and I'll show you little cartoons that are segmented. And they can be labeled. It turns out that the genes for each of those cytochrome P450s are on one TAD or another across 5 chromosomes. We can make pictures of them in 3D. We can analyze them with IC. You know, we can understand when the chromatin opens what histone marks are involved. This can all be measured now. Okay? It's not the bare DNA that we're measuring. There is no bare DNA. It's all wrapped up in chromatin. Why? Because you have 2 meters of DNA in each cell that's 10 microns across. It can't fit in there. It's a charged molecule. You have to neutralize it by these histone proteins and then wrap it up on the nucleosome, which folds it down 7-fold, and then wrap it in the cross-linker fiber, which folds it down another 50x so you can fit it in. There's a higher order, there's a big higher order structure within the nucleus. So in bioinformatics, see, we think of the DNA as one strand, and you could do sequence measurements and say, that's, you know, 5 megabases down there. But in 3 dimensions, it might be right next to one another that you can measure now with the high C that Adika does in the imaging. So we can start to understand the 3-dimensional architecture of this. We can start to see the topological domains. And we can break open the entire structures within the nucleus of a cell. You see, look, you get enhancers and promoters and LADs and pore complexes and regions between the TADs where gene fusion events can occur. This is the thing that's driving the biology, is the 3-dimensional and 4-dimensional architecture of the chromatin in the nucleus. That's what you have to understand. You know, different loops, you have enhancer, you have these CTCF proteins and cohesin like a paper clip, and the variants that we're actually measuring are in the enhancers and the promoters. They're not in the actual gene, which is down there. There's some. So we actually did, and then you can look in 3 dimensions at the transcription within the 4D nucleome context, and you can read it out using the high C technology, which has its own hierarchy that you can read out, see? The one thing that really woke me up was this paper back in 2013, not that long ago, that said, here are those exon coding variants for the cytochrome P450 series. And Professor Zonger, Louis Zonger, said, look at all the non-coding variants in the cytochrome P450. So I went out there and I tried to figure out where they were. What genes did they regulate, and where were they in the 4D nucleome? It invented a new term called the pharmacoepigenome. Okay? The active non-coding domain of the human genome that consists of spatial, temporal, and mechanical regulatory mechanisms of gene regulation that responds to the xenobiotic stimuli. Rewriting the book, the 2nd edition of Pharmacogenomics, by using the things that we learn about the 4D nucleome. Okay? So the experiments that I did to figure this out was I took every candidate variant out of every genome-wide association study or association study in the entire library. Anything that was done that measured a bunch of patients responding to a drug and measured across the entire genome and looked at linkage disequilibrium and genome-wide associations. I took everything out of every catalog publicly available in the world and jammed it into a machine that we built to classify where those were on the genomes against the looping structures that were in the chromatin, and then map the pathways. It's big science. But that's the kind of work I do. So there's the association results. I take them through these boxes: permissive candidacy, regulatory function, variant function, target genes, network analysis. This is the first model that I did. Here's something later called the pharmacoepigenomics informatics pipeline that we created. There's a bigger sample there, the coding variants. There are the non-coding variants, association results, candidate genes, master files. Read them out into pathways and assign the genes into the pathways. Okay, pulling down, yeah.
Can big data make this quicker?
This is big data.
Oh, this is.
This is it, we're there. We're taking down hundreds of studies to do this. Yeah. And all these things down here, chromatin marks, DNA, cipher sensitivity, all this, these are big databases themselves. Yeah, great question. Absolutely. This is big data. This is why they gave me the job, I guess. Okay? And what I found out was that only a little fraction, like 3-5%, a little fraction of the marks, okay, for all these psychiatric studies with the drugs, were coding variants. The rest are introns or intergenic variants in the promoter or the enhancers. It was a big finding back in 2015 when I did the first test, and other people were finding it. In fact, one young guy that I hired, Alan Boyle, is over in Michigan. He invented the whole thought in the first place. They called it the Regulome. He was at Stanford. I bought it and brought him over to Michigan. And I hired a whole team. It wasn't just a geek. I hired 5 of these guys. And here's how it broke down. I did it manually, then I compared it to the epigenomic roadmap, and I assigned whether it was enhancer or promoter, where it was. It was all done by hand at first. What tissue file it was related to, before I coded it up automatically. It spent 3 years in the laboratory just trying to figure out the mechanisms and then coded it up to run at scale, see? Which is the way you do it. Transcription factors, and then related it to addiction, analgesia, which is pain, fighting pain, drug response, including psycho, adverse events, organ injury. Sort of classify it in terms of what kinds of biological effects the drugs were going after and what genes and what variants were involved, and then relating them to what proteins and what pathways. See? You can do it. This is what bioinformatics is for. All right, now, what I did here was, this is just kind of the noise floor of variants that were to assign the disease. And up in here, this is the effect size versus the allelic frequency. High effect size, everybody gets it. So these are really interesting because this is a drug for carbamazepine, which is given for certain kinds of epilepsy and schizophrenia and psychotic events. But what happens is if you have those single nucleotide polymorphisms in the regulatory region, you get something called Stevens-Johnson syndrome. You ever heard of that? You have? It's terrible. Yeah, your skin falls off, it'll kill you. It's a lethal variant. In Singapore and in Taiwan, they have a genetic test that if you give that drug, you have to test for those things. Because if you have them, you'll kill the patient. All right? And but these are the kind of effect sizes and allelic frequencies you'd see, in the noise floor. When you see effect sizes up in here, that's something rare. And it turns out pharmacogenomic marks, both the pharmacokinetic marks and the pharmacodynamic marks, we haven't evolved. Maybe for morphine or cannabis or tobacco or something like that. We could get into that story, but we haven't evolved against all these drugs. We don't have ways to fight those up, so the effect sizes are huge. which is actually pretty good. This is why the drug works. But there are many of them are associated with adverse events and things like that. You've got to weed those out. And most of these marks are pharmacodynamic. We needed those. So this is really big progress. And then the thing that was really neat that we found, and here's an example, is that you'd see on one gene you'd see a mark, and that would tell you about the regulation of the gene that encoded for the metabolism of the drug. But somewhere else on another chromosome that was actually close by in three-dimensional space, there was another interaction called trans that controlled the adverse events. So it turns out that instead of just looking at a genetic mark for the disease metabolism, in three-dimensional space there's 3 marks. One for the disease, one for how you metabolize the drug, and one for the adverse event. You can pull those down and you can change the world. And I said I need a test for lithium. My sister has it. She has bipolar disease. Bad side effects, so, and runs in the family, so can we get a test for lithium? Turns out we can. And we discovered a pathway we discovered a pathway called the glutaminergic pathway, where lithium interacts with 12 genes in one pathway. And what we figured out was that some genes cause different responses in different patients and different adverse events in different patients. And so if you can make a measurement of the patient, what genes they have, you can determine not only whether they would respond to this drug or not, but what adverse events they would have. That would really help. And then on top of it, you can see the trans interactions that are related to those adverse events. And later on, what you can do is you can come up with something called the lithium super pathway that maps onto the different, that shows the neurogenesis, the mania, and the bipolar control, the circadian rhythmicity related to sleep, drug-induced tremor. You can actually, get the pathways and you can read them out so that you can understand the different, the superfamily and the different subfamilies that are related to different metabolic responses and adverse events. This is going to change the entire, so what we need is some math behind it. We need network biology and math to understand these different levels, okay? And then other people, not just our work, replicate your work, which is good. Now, here's something else. Change of gears, but related. Let's say we could go into the electronic health record and mine all the health records for, does this patient have bipolar disease? Does this patient take lithium? Did this patient have adverse events to lithium? Let's just look and see. So we built a system, one of those other jobs that I had that you were talking about. I had to build a system, right, for the hospital, where you take all the data from all the records in the hospital. Here's another example of big data, right? So we have what? 58,000 inpatient visits a year, 2.5 million outpatient ambulatory visits. We've had 20 years of data, put it all in the system. It's all based on diagnostic codes. You have all that data out there, and then what you want to do is do queries and pull all the bipolar patients to see what kind of adverse events they have, see? So this is read out right from, you can see the ethnicity, the age, what kind of insurance they have, mild depression, there's bipolar disease, associated disorders. See, this is the kind of thing we're going to do all over. We put electronic health records in all the hospitals across the world. We pull all this data, we can actually look and see. And then we can relate that to the molecular phenotype. All right? What kind of other conditions they have, like cardiac arrhythmias. Take a look at this. So people with bipolar disease, look at that. Half of them have cardiac arrhythmias. What is that all about? That's a drug adverse event. It's what's driving that. See, then what you can do is that other neurological, obesity, weight, obesity. Look at the numbers over here. Drug abuse, alcohol. See, you can get comorbidities, smoking. Like, schizophrenia, they all smoke. I mean, you can pull that out. Well, what does that have to do with it? Well, if you're taking nicotine, then you have to change the dose of these drugs by 40%. See? You can read it out of the record. So I found 9,226 bipolar patients. You know, in my EMR, and 38% of them had long QT syndrome. Heart issues that were related likely to drugs that they were on. See, this is so we can take it from the bottom up with the 4D nucleome and measure determine the molecular phenotype, and then from the top down, we can go into the record with the big data and we can see what's in there, and we can see then, you see, if you have blood from these patients, they consented to give you the blood, you can get the DNA and you can sequence them, and then you can look for those variants and you can look for those adverse event marks, and then you can relate those patients to the ones that have the various adverse events. See? So I'm just starting to do that now. I got enough in the database so that I can do that. So we're doing it. And look at what you might do. You can actually take a data set, you can genotype the patients, relate them to other conditions they have, use a support vector machine or machine learning, and before you know it, you're likely, my hypothesis is, I could probably figure out once I get a training set, I think I can assign the metabolic pharmacokinetic status by just looking at the electronic records and not even having to make a molecular measurement. Okay? The world's going to change. Let me show you another. It's getting late, but let me show you a couple more stories. Valproic acid. Why was I interested in that drug? Well, sometimes in bipolar mania, it's the drug of choice. Okay? So I'm trying to develop a panel of all the different drugs you take for bipolar disease so I can get a genetic test out on the market, into clinical trials on the market, help these patients. Well, here is, look at this for these two pigs had a brain injury in the laboratory. Bad brain injury, traumatic brain injury. All right, TBI. All right? The pig that got normal saline plus valproic acid looks pretty good. Can run over and get the food. The one that didn't have the valproic acid is nearly dead. One dose. Did that experiment 35 times. You know, and you can measure the neurological industry severity score. And you can look, and you can see that normal saline, the severity score was much higher at peak, and then disappeared faster if they had valproate. Okay? Wasn't as bad. One dose. So you say, what's going on? This is like the beginning of something that's changing, like it's starting to change the whole field of psychiatry right now. Okay? I'm not going to show you the movie because it doesn't run. But anyways, that poor guy couldn't find the food, so you don't want to see. All right? But here's the thing. You know, by the way, picture smart, so you can do neurocognitive testing and you can see that the valproic acid really helps. So the question is, what's going on? All right, there was the injury. You can look at the tissue. There's your data. There's an early cartoon of my pipeline. And look, some genes are upregulated and some genes are downregulated when you take valproic acid. The ones that are upregulated all have to do with neuronal generation in some ways. This is actually very similar to what Adika's doing with his like Yamanaka factors. This NeuroD1 is a Yamanaka factor for the brain. It's the same darn thing. I got my labs next door to the guy. And look at down here, it's glial cells. You have two kinds of cells in the brain. Huh? You got the neurons, you got the glial. The glial support the neurons, right? And what's going on? One's upregulated, the neurons, and one's all downregulated, that's the glial cells. You got neuroregeneration. It's regenerating your neurons in your brain. How does it do that? Valproic acid, it's NeuroD1, which is a master transcription regulator, and upregulates 300 genes that turn on neurons and downregulates a bunch of genes that turn off glial cells. It's incredible. Nobody ever knew what it was. Drug's been on the market for 45 years. Nobody had a way, knew how it worked. And nobody knew about the variants that you could measure to adjust the dose or see whether you're going to respond or not. And look where they're all at. And the answer is the promoters. They're not in the gene, they're in the loops that control the expression of the gene that you can only learn about by the 4D nucleome. And look at one, this is called the NMDAR receptor, which is a big deal in neurobiology. And you'll have one cis interaction, a bunch of trans interactions. You know, what happens is you know, a chromatin is in an open chromatin region, that changes first. Then a bunch of trans interactions occur, and it cascades a bunch of different biological mechanisms that regenerate your brain. That's why the pig can walk around. You'd never figure it out without the 4D nucleome. And then you can make mechanisms. How does it work? And then you could do something else. You could look at the cells and you could say, what's going on inside the cell? And what you can see, the hypothesis, which we've now proven, and I'm running out of time, is that, as Aditya had talked about, the stem cell-like nucleus is round, the terminal differentiated nucleus of the neuron that's terminally differentiated is oval. It's ellipsoidal. So what happens is, it's called the mechanogenomic hypothesis, that you have all the chromatin is in its stem cell-like spherical state. You hit it with a drug and it terminally differentiates the cells and the nucleus changes and all the interactions between the TADs change. And you can actually make measurements of that. You can actually enhance your methods to measure your chromatin capture data, right? Where you have the chromosomes and you've seen this data. And then if you change the size, all that was done with binning that had uniform bin sizes. How about if you change the bin size in your chromatin capture to like look at the features that you're looking for, adjusted for the size of the tags and the elements that is in the 3D space. You can improve the resolution. And then you can use an algorithm to assign the enhancers and promoters for different drugs. Here's two, ketamine, which we're using to treat for depression, that acts just like valproic acid. The valproic acid, they both regenerate neurons. And what you can do is you can read out all the different tags. You can get a readout, and this is the Dixon numbers, on different chromosomes. You can read out all the different tags, chromosome 4, 7, 9, 10. This is where these tags are for the different drugs, different pieces along the way. And you can get a three-dimensional map inside the nucleus, all the different chromosome territory areas that are turned on by the drug all at once. And what you can start to do is image those. So here's in like one chromosome 22, there's proof that you can image all of the tags. So imagine a day, which is going to come real soon, where you have a fluorescence-activated cell sorting machine that has microcomputer tomography. So you take all the nuclei, all the cells, one by one, you sort them. This is a glial cell, this is a neuron. This has a spherical nucleus. This has an oblong nucleus. And then you put the fluorescent markers in there and you show what every TAD is, 3 dimensions. Think about it. Every one of us would have a different profile. And we can individualize our understanding of which genes, which drugs you can take. It's going to happen. You guys could do it. It's the power of an idea. So here's a picture. So look at what happens actually. This is a new paper I just put together with my collaborator. Okay? Here, no drug. You take no drug, you have the TAD boundaries over here, the regions between the TAD. Then you take a drug, a psychoactive drug, what happens? the chromatin changes in the TAD, in the gene expression within the TADs change. It's the TAD, the topological domain within the chromosome territory, which has the genes and the gene sets that code for the different pathways, which is the functional unit of the chromatin. It is what you got to read out. And you can also determine at the boundaries the most significant SNPs that are likely causal for the effects that you're looking for. This is all new. Okay? It's all been discovered in the last 2 years in the laboratory. Just imagine what you could do with this. I think I can say by the look in some of your faces that you are imagining. You can industrialize the process of assigning any biological function through labeling, through chromatin capture, through imaging, and through high throughput and big data. And you can individualize the assignment of a different biological effect using this capability. And you can discover new drugs. You can see that this drug, which works for epileptics and mania and now traumatic brain, you can see how it works. You can see the mechanism of exactly how it works. That we didn't know. We just took the drug and it worked. But some people, like if you're pregnant and you take the drug and kill the baby. You don't want to take valproic acid if you're pregnant. You don't want to take valproic acid if you have those SNPs that look like those Stephen Johnson SNPs. So you have to look. You have to have a test. See? Don't put a pill in your mouth until you know whether you can metabolize that pill or not right. And we're inventing the way to do that by applying the 4D nucleome concept. There's no way that you could read this off of a piece of DNA through a sequence machine alone. It's impossible. This is how biology, bioinformatics, computational biology, and the big data stuff is going to work. And then you can, I got another whole talk, run out of time to show you how you can, I did the three-dimensional study with the machine learning on the cells. Suffice it to say, you can throw another module into your PIP block to measure the mechanico-genomic effect through the imaging and throw it in there to increase the fidelity of your assignments. Okay? So, what does the future look like? Well, individual comes in and you measure their genes and you measure their electronic health record and their physiome. You take some socioeconomic and demographic data and you try to see whether they've been exposed to anything in the environment, other pills or chemicals or whatever. And you mash that up in a big data aspect to establish a pharmacological phenotype from the top down and a pharmacoepigenomic phenotype from the bottom up. So you move from the first generation that Dr. Mrazek invented and that we put onto the market. We move to the second and third generation and combine them. Okay? And then what you can do is you can load this into a system and get a training sets going, put it through a machine learning, and you can assign the pharmacological phenotypes, and you can assign new pathways and new understanding for how the drugs work. You can change the way that we understand the pharmaceutical industry and the mechanism of drug interaction in a patient and tissue-specific manner. So, gene co-expression modules can be defined. This is like the way we did our work. And then when we applied the 4D nucleome concept, we changed the paradigm of how this whole field of pharmacogenomics, measurement, and pharmacologic discovery occurs. You know, just look at gene co-expression. Now you look at spatial, temporal, and biomechanical dimensions of the non-coding genome and the concept of the transcriptome on those levels. Okay, pulled together by the 4D nucleosome concept with the understanding of the chromatin architecture. CNS drug targets. dump a drug on a petri dish and see how a neuron responds and do a patch clamp, and you know, it's done all over the world. Old-time neurophysiology, it's going out the window. Most drug targets extend beyond the small number of candidate molecules involved in signal. Researchers can now investigate the 95% of the transcriptome of the human brain that nobody's ever looked at. Okay? Preclinical screening. We used to have, still does, pharmaceutical industry have industrial big robots. You take every drug that you got in the world, you put them in the big robots, put the cells down there. You seen it? Forget it. It's not how you're going to do it. You use predictive modeling and the deep learning, and you build your candidates that way and use the bioinformatics and silico to do it, and then you do your experiment. You're wasting all that money and all those robots and all those cells and all those drugs for nothing. You need to start with a mechanism and a model before you can start to refine it. Epigenomic drug discovery. We used to focus on single molecules, readers, writers, and erasers. Now we're looking at the entire complex of them across the whole human genome. Okay? Pharmacogenomics. Imagine this, I learned this way back, my first consulting gig. I helped this whole guy, my buddy, Felix, was head of pathology for Warner Lambert Park Davis, the company that brought Lipitor, you know, the company that discovered Rogaine, Viagra. Right? They had a drug called Resilin. It was a really good drug. Maybe if you were older guys remember that drug. It was a great drug for diabetes, okay? Except for the 90 people it killed after it was released out on the market. So the question is, why did the Resilin kill these people? Well, if you don't use pharmacogenomics to stratify the patients, you know, there's no one drug that everybody can take. What we have to do is, in the drug development process, we have to stratify the population for ethnicity. For example, there's a lot of the best markers for bipolar disease, and some of the other things are in the Han Chinese genome. Studied that extensively and published papers on it. Respond completely different to these drugs. So if you're giving them here in China, it's going to be different than what you do with in Africa or North America or the European or something like that. Each ethnicity. So genders are different. You know, androgen is different than the female hormones, etc. This all has to be brought in. It can be understood in the TAD concept. Off-target effects and adverse events. Mitigation using systems pharmacology. That is a sexy idea. A lot of good value to that. Okay, I helped put some of that together myself with the FDA and so on. But most adverse events are mediated through interchromosomal contacts in the trans. Okay, with transcriptional hub. Disease risk variants are located within the same networks as drug response and adverse event variants. And so, by using the 4D nucleome concept, and the idea of cis regulation, close and far, and trans regulation across the TADs, we can rewrite the book on how pharmacogenomics and drug discovery works. That's what I've been doing for the last 10 years of my research life when I have time to do it for my other duties. And it's just one example of what we can do with some of the things you're learning about in this institute. So I'm delighted to be here, Professor. Thank you for having me, and Michael. And if there are any questions, I can answer them now. Okay?
Question?
Yeah.
I got a couple of questions. One, does it matter the pH levels in the body?
Yeah, it does. Yeah, it absolutely does. There's what I don't understand is, what really matters and what I didn't have time to explain is what local chemical, environments, and pH would be a big one because it would change all the association constants. And there is good evidence of different kinds of pH in different tissues. But one of the things that happened with the epigenomic roadmap and the GTACs that we use in our assignments is we have actually a master tissue file. And like for one of the things that we do is we use the Allen Brain Atlas, and there's also, I guess, in China, I haven't been able to really have time to look into it, but there's another brain atlas where they have transcriptomic, whole brain transcriptomic data. We look at the different neuronals, the different circuits within the brain that are related to a different kind of thing. And we also look at the tissue specificity of the response. Each diseases have their own different tissues where the genes are actually activated and the chromatin is turned on and off and affected by the drugs and so on and so forth, like diabetes. The pancreas is an obvious one, the adipose tissue, the spleen, you know, actually the spinal cord is involved with that. So for each disease or condition, there's a different set. Like in depression, for example, recently we've been looking at, it turns out that likely depression has 4 different neuronal circuits in your brain, nuclei circuits, different brain circuits that are involved in depression. Each one of those, we call them now different biotypes. And so in the new world of depression, say in 5 years or so, you either have 2 kinds of depression. You're just starting out. You have brand new depression. Depression is a chronic disease. It gets worse and worse. It's neurodevelopmental, and by the time you're 20-something, if you have it, it's a chronic disease. You're never going to get well again. It's brain damage. If you don't treat it, your brain will get worse and worse. So you got to stop. So it turns out that you're and the name of the game is to get you out of depression, major depression, as quickly as possible. And it looks like there's 4 different biotypes, 4 different circuits in your brain. And likely what's going to happen is you're going to take a drug, ketamine, which I've been studying a lot. I just referred to it. I got a whole nother talk on that one. And ketamine's like a miracle. You're profoundly depressed. You take ketamine. Some patients just come out of it like immediately. It's incredible. All right? And we're trying to figure out which people can handle ketamine and which can't. Turns out when you put people into a brain scanner, there's a part of your brain called the subgeniculate cortex, which is activated by ketamine. And 3 of the 4 biotypes are circuits that we've studied. Ketamine actually the subgeniculate cortex isn't turned on. Biotype 3 it is. So our hypothesis, those people with that circuit, ketamine would not help, it would in fact would hurt. Okay? Now, there's something called transcranial magnetic stimulation. So this is pulse sequences, magnetic pulse sequences that you can put in different parts of your head to aim at those 4 circuits. And what you can do is actually electrically pulse them back to the starting point and get rid of the depression pretty fast. Another thing for profound depression is electroconvulsive therapy, which is like 4th generation familial electroshock therapy. Actually works great now, very safe. And so likely what's going to happen in the future is if you have major depression, what will happen is we'll make a measurement of the different biotypes in your brain. When we're going to give you ketamine, we're going to give you transcranial magnetic stimulation, we're going to give you ACT, and we're going to get you out real fast, and then we're going to put you on a drug to maintain it. That's what's going to happen. It'll revolutionize the field. And there was just a study that there's these 55 drugs, but likely only 6 or 7 of them are any good. So we're going to down select the antidepressants a lot. And so I've got a ketamine super pathway with what happens. Ketamine is fascinating, actually. Another drug that might pull you out of depression, which is something that Cary Grant used it and so on, and are LSD and psychotropic drugs will likely be also used in small doses. Ketamine is actually very interesting because it's an antidepressant at a certain dose. It's a hallucinogenic drug called Special K, which is a street drug at just another dose. It's an anesthesia that's used in most of the time when you go into general anesthesia, they give it to you. So depending on the dose. So one of the things that in the first generation test, it was adverse event avoidance. And then third generation tests that were, so certainly the second generation with pharmacoepigenome, we can read out what kind of dose you should take by looking at the genetic marks and then looking at a table of different things that modulate the dose. Give you an example, ketamine. You would think that if somebody walked into your clinic with a huge body mass index, obese person, would they use more of the drug or less of the drug? If you had somebody really big, your intuition would be give them more drug, right? They're bigger. Well, ketamine is soluble in the adipose tissue in the fat. So it's stored in the fat. So if you give ketamine to somebody who's obese, it actually releases the drug over a longer period of time. Because the pharmacokinetic profile, there's only really one of the cytochrome P450s that metabolize ketamine. CYP2D6. And there's a 2,000-fold dynamic range of how that enzyme works. And the biggest the biggest modulator is whether you're a man or a woman, and whether you're, have estradiol or not, and you have female hormone, and it changes the dose, it changes the activity of CYP2B6 into a ultra metabolizer, and it'll remove all the ketamine from your body in a week. So it's all about, so we need a pharmacokinetic measurement model, which is not so much about the different cytochromes, is what is your body mass index? What hormones do you have flowing through your blood? This can all be measured. So when you walk into the clinic in another 5 years, we're going to be able to, 5 or 10 years, we're going to be able to tell you what dose of these drugs you ought to take and where you ought to start. Yeah?
I would like to ask that you're saying that after the 50s that we didn't have like the new drugs. So that what's your guess? It seems like you are more focused on how to like focus on targeting the drugs on the specific targets. But what about developing new drugs? I think that you haven't mentioned about that. So could you comment on
Well, by the way, thank you for the question. I was referring to drugs in the central nervous system that help psychiatric patients or patients with other neurological diseases. That's where the problem in the drug discovery pipeline is, in those neurological and neuropsychiatric drugs. Yeah, there's a lot of good drug development starting to go on. Some of it is actually informed by the pharmacoepigenome. Like take histone deacetylases, for example. Valproic acid is actually a histone deacetylase, as you might know. So what does that do? So one of the reasons that the nucleus changes its shape is that the first thing that happens with these drugs is the chromatin is attacked. The histone deacetylase, when the histones are decetylase, the euchromatin turns to heterochromatin and it compacts. And it turns off the biological effect. So, and there are second generation histone acetylases which are much more specific. Valproic acid hits every nucleosome and causes all the chromatin to expand or contract, depending on its presence or axis. It's like a sledgehammer to your chromatin in its overall organization in terms of U and heterochromatin. But these targeted deacetylases, for example, a friend of mine is working on some that would affect a certain family of histones in a tissue like your gut, where you have leaky gut syndrome, which is like modulated by an epigenomic. So I think you're going to see a lot of drugs that are going to be more specifically targeted at different aspects of the chromatin structure. And and some of those drugs are being developed right now, and I think we have the ability to understand exquisitely their mechanism with this approach. Yeah, it's a really good question. And that's just one example. Yeah.
Thank you, Professor Huang. Indeed, is a genomic difference between individuals makes our view different. So that's that's your talk, main ideas about the genetic difference because different people have different reflections or results or if they take the medicine. So I think most of the medicine, maybe many drugs are taken orally, right?
Yeah, by the way, this is really important, your point.
Yeah, that's the very point.
Well, taken orally and taken at certain times of days at certain doses.
Yeah. My question.
However, ketamine is taken, ES ketamine is taken in nasal form.
Okay.
Yeah. And, RS ketamine is given, as a injection into the bloodstream. Yeah.
That's the point. I mean, if the drug is taken orally, so we all know there is another genome in our body, that's the microbiome in our gut.
Yeah, by the way, you're right.
Yeah, there are more and more reports that talk about the microbiome help us to digest and enrich molecular of drugs. So,
no, you're absolutely right.
They do a lot of, scientists already do several research that they have found that the microbiome in the cancer patient influences the result of their immune therapy, right? Such like PD-L1.
You're absolutely right. You're absolutely right. So, and then one of the things that's crazy as well, especially for pediatric patients, to build on your theme, is, we just have pills in certain sizes. But, depending on your weight, depending on your age,
yes.
What we should do, and we have the ability to do it, is you come into the clinic and we make a measurement and we figure the exact dose that you need, push a button and get the pill made, and then we figure out what time you ought to take it, how often you have to take it, and whether you should take it orally. For example, if you're going after something, for irritable bowel syndrome or IBS-D or something like this, it's not clear, for the reasons that you mentioned with the different microbiome signatures and so on, that would be a smart way to go. I totally agree. And that would be other boxes that you could put into the model, depending on what kind of metabolic pathway. Yeah, absolutely. And the other thing that's really good about your idea is that it focuses more on the tissue specificity of the drug and where it's acting. In the first generation, I just look at the liver. Like, for example, in the CYP2B6 example with ketamine, it's most likely that most of the metabolism is with cytochrome P452B6, that it is in the tissue of the neuronal circuit itself. It is metabolized in the brain, not in the liver, by the cytochrome. So we need, but we can make a simple compartmentalized model. So you have four tissues. So you have the human body, you have the different tissue, you have the brain, you have the brain circuit, you have the liver, you have, say, the spleen, whatever it is, you just make a model. And if you want to include the microbiome, because the thing is going in there and it's being broken up in the stomach and then going into the bloodstream, you stick it in your model. You can do it. Absolutely. There's a guy named Larry Lesko in Florida. Look him up. I Google him. He knows how to do it. You can read his book.
Oh, thank you.
Yeah. Excellent question.
Maybe we should be adjourning to the coffee hour.
I just burned through the coffee hour. Yeah. I'm sorry, Professor. I talked to you. Yeah.
In this world we live in today, where we live in this big chemical soup,
yes.
Chemicals coming all the time.
Yes.
With this exciting new research, you'll be able to identify all the chemicals and all the interactions.
Well, it's a huge problem, but, so you want to start off, I think the way to do it is just make a list and hit the bad ones first. But one thing I didn't talk about, and I'm still trying to figure it all out right now, is that each of the cytochrome P450s are on different chromosome territories at different TADs. Each one does describe a different metabolic pathway, some of which are for so-called xenobiotic substances or environmental substances, like polyaromatic hydrocarbons or something like that. So we could zoom in. And then we do have evidence from epidemiological records of where we had EPA Superfund sites in the United States with lots of, you know, PBBs or something like that, that you can see in hand, you know, big upticks in certain kinds of leukemia or different kinds of blood cancers or something like that. And you can start to sort it out. But it's well taken. I mean, and then I cut a bunch of slides out, but one of the things that we really need to do now is you think of genes by environment using the phenotype. So I mean, I suppose probably all we can get out of most of what we can get out of the genomic term by looking at this hierarchy of the epigenome. But there's still, like the gene by environment interactions, the environmental interactions is where the money is right now. And in order to get the assignments for the decision support above 50%, you've got to go into those cross environmental interactions and measure something about the environment. It's very important. Yeah, it's a good question. It's definitely an area for research. Can be done.
Well, let's thank you for this